doing we're seeing notes on ourselves.
Okay, ready to move on. Our next presentation, and I don't envy him following that last talk, will be Robert Lee. Rob is an associate professor of Otorhinolaryngology and Physiology in the Perelman School of Medicine at Penn. Rob.
So I am in ENT. Seem to be. There we go. It is a true pleasure to be here. And I just want to say that, so I am trained as an epithelial physiologist actually in Kevin's lab where I was a PhD student. I started as a postdoc in Noam Cohen's lab, who you heard talk earlier. I never in my life thought I would work on taste receptors. But while I was a postdoc in Noam's lab, that's when Manel approached, and Bob and Danny Reed and other faculty at Manel approached Noam to start working on taste receptors in airway cilia. And what I thought was a side project, eventually blossomed to kind of dominate my career for the last 15 years and has led to a really productive and amazing career and collaborations and interactions. And I don't know what my career would look like otherwise, but I do know that I'm incredibly grateful for the role that Bob and other Manel faculty like Danny Reed and Gary Beecham played in this process. And it's been a really fun ride, and it is really a pleasure to be here at this symposium to honor Bob. And to start, I'm going to take a slide that I have used many times over the years that I have actually stolen from a review that Bob wrote, kind of modified it a little bit. But I thought it was appropriate to start. I had to go back and look at which review actually came from, because back in the early days when Noam and I were trying to figure out what taste receptors did, I read a lot of Bob's reviews. But we know there are 5 canonical tastes, and I don't have to go through this for this audience, but we know that bitter, sweet, and umami are encoded by these 2 families of G protein coupled receptors, TAS1Rs or T2Rs and TAS1Rs or T1Rs. The dogma in evolutionary biology and in taste is that T2Rs or bitter receptors on the tongue protect you from poisons. Many of the most toxic products we know are bitter plant products that activate T2Rs, while T1Rs on the tongue allow you to perceive nutrients like sugars and amino acids. We know, however, from work that Tom Finger has done, also work that Noam and I have done and that I've carried forward in my lab, that both bitter and sweet receptors also detect bacteria. Acyl homoserine lactones, which you heard a lot about during this talk, activate various T2Rs. A former postbac student in my lab worked on Pseudomonas quinolone signals that activate several isoforms of bitter receptors. One of the first people who worked in my lab, Ben Hariri, who's now an ENT, worked on how staph D amino acids, a lot of D stereoisomer amino acids are sweet, activate T1Rs in the airway as well. We know from work from Prashant Chelkani's lab and others that staph-competent stimulating peptides also activate T2Rs. And so these are clearly not just taste receptors, they're chemosensory receptors that we use for taste, but also use for other functions. And we know there are probably endogenous molecules that we don't even know about that activate them. And that's probably the reason why these receptors are expressed all over your body, right? And a major question, despite the fact that we've known they're expressed all over your body for decades, is still, what do these extraoral receptors do and what activates them? And this may take another 20 or 30 years to figure out, as we're still discovering cells where they're expressed where we didn't know they were expressed. I have spent the last 15 years working with Noam and other people in rhinology and at Penn, working on this in the context of the nose and sinuses. I've also started working recently with my colleague and collaborator, Ryan Carey, who's there, on how bitter receptors in the oral cavity also regulate responses and actually are involved in oral cancer. For the purposes of this talk, though, I'm largely going to talk about the airway epithelium. The airway epithelium, as Noam said, and Noam's talk actually set me up really well for an easy go. The airway epithelium is lined with mucus. The purpose of this mucus is to trap microbes, particulates, pathogens, all the bad stuff that you breathe in gets stuck in the mucus that lines your airway. Goblet cells in your airway secrete this mucus. Ciliated cells, through regulation of ciliary beating, regulation of fluid homeostasis to keep the viscosity of this mucus appropriate, clear this mucus. And so while you are sitting here, you are constantly clearing mucus from your nose back to your throat, and you're constantly clearing mucus from your lung up to your throat, where you are swallowing and getting rid of all the bad stuff that you're breathing in. Because of that, and because of the structure of your airway, which is the most predominant cell being these ciliated cells, these are really the first point of contact for pathogens, for irritants, for particulates. This is a slice of middle turbinate that was embedded in an auger that a former postdoc from my lab stained for cilia in collaboration with Mike Kohansky, who's going to talk later. And you can see here at the apical epithelium all of these cilia that line the tissue. We study these cilia and we study these cells in air-liquid interface cultures, exactly what Noam talked about in his talk. We take residual human sinonasal tissue, we dissociate and culture basal cells, we differentiate them in air-liquid interface, the same model that we've used for a lot of these studies. We get formation of nice cilia, we get formation of tight junctions, which you can see here stained with phalloidin. Let's see if I can change this to a laser pointer. You can see stained here with phalloidin. And you can see that we've got these cultures that almost are a tissue type culture. We can do all kinds of imaging on them. We can do transport studies. We can do electrical studies because they form electrical resistance. It's actually a really great and productive culture model. And it's a translational model because it's been used, for example, in cystic fibrosis to help the development of CFTR correctors and potentiators that have really revolutionized treatment of this disease. We studied for many years, as Noam mentioned, bitter receptors in nasal modal cilia. We found a number of different isoforms like T2R38, T2R14, and we characterized a pathway by which bitter products, bacterial products, bitter drugs can stimulate these receptors in nasal modal cilia and lead to this pathway that Noam talked about, where you've got calcium activation of nitric oxide synthase, which can lead to direct bacterial killing, as well as ciliary beating through protein kinase G. And we've characterized a lot of the molecular pathways that are involved. We've characterized a lot of the regulation of this pathway by other G protein coupled receptors as well. This pathway is actually really important and really unique because of all of the G protein coupled receptors that we've studied in these cells. These are the only receptors that activate these robust NO responses. And I think that's because of the really close proximity of cilia to eNOS, to the endothelial nitric oxide synthase isoform in these cells, which is a really important component of this transduction pathway. And I think that what really needs to be highlighted is that in all tissues, in many tissues, these bitter receptors are incredible opportunities for drug repurposing. In our oral cancer work, a former grad student of mine, Zoe Miller, showed that we can use lidocaine to activate T2R14 to activate apoptosis in head and neck cancer cells because the T2Rs are coupled to apoptosis in oral cancer cells. This has led to a clinical trial that Ryan Carey's running to look at patients injecting lidocaine into their tumors to see if this is useful in their cancer. And we can take information from that and we can extrapolate it to the airway as well. Because if we look at our airway ciliated cells and we stimulate them with lidocaine, we can see a pretty robust increase in ciliary beating. And this is through activation of T2R14. And so I think that in all of our studies of extraoral taste receptors, the idea of agonists and the idea of drug repurposing is very sort of cross-pollinating. And I think that as a field, we can really learn a lot from each other in the work that each other does. We also did some drug repurposing studies looking at diphenhydramine, which is a classic H1 antihistamine that is also a really good activator of T2R14 that can induce pretty robust ciliary beat frequency responses. And so we have investigated the molecular details of this pathway And our colleagues, Noam and other physicians at Penn, have really investigated the disease-related aspects of these beta receptors, right? And Noam talked about this in great detail. Patients with non-functional T2R38 and cilia have increased gram-negative upper respiratory infections. This was the first thing that we showed almost 15 years ago now. They have increased biofilm-forming nasal bacteria. They have increased susceptibility to chronic rhinosinusitis. They have decreased outcomes after sinus surgery, which Noam talked about. We published a paper a few years ago that actually, others have published that people with cystic fibrosis. So in cystic fibrosis, people have an innate susceptibility to gram-negative respiratory infection. Bacteria like Pseudomonas aeruginosa, Klebsiella pneumoniae, and others are classic bugs that infect these patients. People have shown that if you have AVI T2R38 or the non-functional T2R38, you have an increased risk of being colonized by these bacteria before the age of 14. We showed a couple of years ago that actually this T2RNO production is generally impaired in CF nasal epithelial cells because of a defect in CAM kinase phosphorylation of eNOS, which we think actually contributes to their inherent susceptibility to these infections. And so I think that we're still kind of understanding the role of these beta receptors in upper respiratory infection and what diseases they're going to be important. But I think that all of this clinical data underscores that learning what other receptors are in cilia is important because they may also be clinically relevant as well. We've been asking over the years what other chemoreceptors are in motile cilia so that we can understand what they do and what activates them. We know that calcium sensing receptor, the extracellular calcium sensing receptor, is a really good marker of airway cell cilia. Leon, my postdoc, who I talked about before, spent a lot of time trying to figure out what it does. Still haven't done that yet. But one of the receptors that is in the same family that we also believe is in airway cilia that we think is important, and we think we do know what it is T1R3, the subunit that is in the sweet and umami taste receptor. We have done a number of controls. We initially found T1R3 staining in tuft cells when I was in Noam's lab, but we also noted staining in cilia, which we've tried to figure out over the years what it does. We've done a number of controls that are posted on a preprint that hopefully we'll see the light of day very soon because we just got an R01 funded for this study. We came to the conclusion based on our studies that T1R3 in airway cilia actually regulates airway surface liquid glucose. So in your airway, you have a serosal glucose concentration in a normal, healthy, non-diabetic person of about 5 millimolar. That's your normal blood glucose level, give or take. In your airway epithelium, there's a constant paracellular glucose leak. And the way that your airway surface liquid or your ASL glucose is maintained low is through apical reuptake. And so if you look at the nasal mucus or the nasal secretions of a normal person, the glucose is about 0.5 millimolar versus 5 millimolar, so about 10-fold less on the apical side. And that's mediated through apical GLUT2 and apical GLUT10, while the cells also will uptake glucose through basolateral GLUT1. And the dogma is that this is bad because more airway surface glucose actually results in an increased carbon source for pathogens. So when bacteria get stuck in that mucus, if they have a lot of glucose to eat, they can actually do better. If you restrict their glucose, they can actually do worse. When we looked many years ago at chronic rhinosinusitis patients, we found that chronic rhinosinusitis patients of all types have increased nasal glucose levels. And this is independent of their blood glucose. So it's not just because these patients are diabetic and have higher serosal glucose. When we actually cultured cells from these same patients, when we took them out of their inflammatory environment, when we cultured them under the same conditions, they could all regulate their glucose very, very well. And so we think this is a consequence of the inflammation, but it remains to point, what can we use to help regulate the airway surface liquid glucose in these patients? We found, and have pinned this down to ciliated cells, that if we use the T1R3 agonist sucralose, we can increase the concentrate, the expression levels of GLUT2 and GLUT10 in our nasal ciliated cells. And this is actually sufficient to lower airway surface liquid glucose. There's no effect on sodium glucose cotransporters. And if we look at a protein level, we stimulate our cultures with sucralose versus control, we can see increased levels of GLUT2 at the apical membrane. If we do this in the presence of lactosol, or if we do it in Bob's T1R3 knockout mice, there's actually lower GLUT2 at the apical membrane. So, and we've also shown through functional assays that this is sufficient to actually reduce the glucose in the airway surface liquid. And so we think this is a novel kind of role for a taste receptor in airway cilia. The question that has really held us up is, how does T1R3 signal? We have, looked at so many different types of signaling in these airway epithelial cultures, from calcium to cyclic AMP. T1R3 has really eluded us, but we've got no changes in calcium, cyclic AMP, or NO, no changes in ciliary beat frequency. One thing that we've started to look at is arrestin recruitment to cilia. So most of us think of the G protein phase of G protein coupled receptors, where the dissociation of the G proteins leads to effector signaling. There's also another arm of G protein coupled receptors, which is arrestin signaling, right? After G protein couples dissociate, phosphorylation of the C terminus, sometimes without phosphorylation of the C terminus, arrestins can bind to G protein coupled receptors and cause other downstream signaling, often through ERK or other kinase pathways. We saw very excitingly that with sucrose in our cultures, we can see arrestin recruitment to cilia, which seems to be a very interesting phenomenon. So control, we see very little arrestin in cilia. With lactosol, we see almost nothing. We get some arrestin recruitment to cilia with sucralose, and it's blocked by lactosol, which is really fascinating. We're trying to understand exactly what the pathways involved in the upregulation of glucose transport. And we've shown that actually pharmacological arrestin inhibition also blocks the effects on GLUT2 and GLUT10. We know that this may also be infection related as well, because these sweet D amino acids are produced by nasal bacteria. And so we think this may be a pathway that we can exploit with sucralose, but is also endogenously activated by these nasal D amino acids that you get getting from staph bacteria and not by Pseudomonas bacteria. These are all clinical isolates of staph. And so the pathway that we've kind of worked out, staph secretion of D amino acids, we think activates T1R3, increases glucose transporter expression in the airway, and leads to this innate defense response to sequester airway surface glucose. I'm not going to talk about it because I'm completely out of time. We're also interested in other proteins that are not GPCRs. We've been interested in this one called MS4A8B, which is actually a great marker of airway cilia. It is a protein that has very little homology, except people have proposed to the ORA calcium channel. It's also been proposed, the prototype of the family is this protein called CD20, which has been proposed to be an olfactory receptor in certain types of olfactory neurons in mice. Alex, I'm not going to show the data for lack of time, has shown that this protein is actually a really potent regulator of the ORA calcium channel. We don't know what activates it in cilia, but we think we have an idea of what the signal transduction pathway is. And it's really fascinating what cilia will actually signal, because we know so little about how the cilia signal transduction actually occurs in airway ciliated cells. And so, main takeaways from my talk: extraoral chemosensory receptors serve many functions in airway cilia. We think there's disease importance in chronic rhinosinusitis as well as cystic fibrosis. Airway cilia use both GPCRs and non-canonical receptors. And I think this is a perfect opportunity. And I think the work that Noam and I have done, and I think the work that Ryan and I are doing in oral cancer, it shows that these are perfect opportunities to repurpose or develop new drugs to target and stimulate beneficial therapeutic responses, especially in the ENT space, where we can use topical nasal rinses or sprays to directly target the tissue that we're interested in. And I want to thank everyone in my lab who's contributed to this, everyone in the Carey lab, my collaborators at Penn Rhinology. We've been very fortunate with funding, and special thanks to Bob for all of his support over the years. And my email address is readily available online and also right here if you want to talk about anything, and I'll be here all day.
We have time.
So I'll take the first question. So as I've understood it, essentially the bacteria are secreting these D amino acids to trick the sweet receptor to say, no bacteria around here, right? It's like a trick. Is that my understanding?
So I think that, so, right, there's multiple roles for the sweet taste receptor, and when we start thinking about what it means, it gets complicated because of the stuff in tough cells. We think that the sweet D amino acids are suppressive of the tough cell responses because they're activating the sweet receptor in the tough cells. In the ciliated cells, to me, it almost seems like an immune mechanism because you're stimulating the cells to uptake this glucose to then starve the bacteria of glucose. So I think the bacteria would not want to make the D amino acid so that they have more glucose floating around, but that also would prevent them from suppressing the tough cells and the innate immune responses that are stimulated by the tough cells, which Mike will talk about and other people will talk about later. So I think it's a very complicated question, what's good and what's bad for the bacteria, and may be different in different disease states.
Thank you.
Thank you. Great talk. My question is kind of tangential to the CF nitric oxide reduction. Have you ever looked at PCD and can you ever reconcile why in patients we see decreased nitric oxide production, and that's one way we diagnose it with nasal NO testing, and these receptors? Is there any connection there? I know there's a connection in CF, but I'm just curious if we can ever understand why in PCD it's so low.
So if you can help me get PCD patients, we can look at it. So, and we can do the cultures from nasal brushes, and we have talked about it. And I will say that in rhinology, in 10 years as faculty, I think we've been able to get cells from maybe 2 PCD patients that have come in. And so the barrier to looking at this has been cells. And you're not the first person to ask that question. And the problem, the interesting thing is in cilia, the genes that are regulated, that are important for motility are also the genes that are important for intraflagellar transport and localization of receptors and all this other stuff. So it makes a lot of sense that blocking the motility, especially in dynein arm mutations and things that are going to cause PCD, maybe you're screwing up signaling as well. And so that would make a lot of sense to me, but we've not been able to get enough tissue to actually demonstrate that.
Well, you're in luck. I do brushings almost every Friday, so
That'd be fantastic.
Lisa actually came. We had an IRB with a job. She said, well, she said no. So you can talk to her. I tried this 10 years ago with the BCD clinic with Maureen before Lisa, and then when Lisa came on board, it's got 86. I'm not the one. Ron, quick question.
All right.
What can you try for a question, Kevin?
All right, go ahead.
Timeline for the upregulation of the glutes. How long are you talking?
18 hours. We've only done it overnight, 18 hours.
18 hours. So
Yeah.
if we have our patients snort Splenda, that should work.
So I've been scared to try it, but I've been very thinking that this may be potentially the benefit of xylitol, right? You know, in the literature, xylitol seems to be helpful. I've tried xylitol myself just to see what it is. It does taste kind of sweet. And I'm not sure, maybe Gary or Bob or somebody knows whether xylitol actually activates T1R2 or T1R3. Or maybe Jordan knows. I don't know the answer to that question. And I've been a little bit scared to try it, but I think that this may explain some of the benefits because the xylitol literature is so bad. Like, no, it doesn't seem like there's any real mechanism for why it works. They're claiming that it's, causing a metabolic cycle in bacteria, but I don't know.